Okay, my name is Luis Heriberto Vázquez Mendoza. I'm from Escuela Superior de Medicina, IPN. And the topic of my presenting is repurposing of FDA drugs as potential estrogen receptor beta agonists using multicomplex based pharmacophore maps, a new approach in breast cancer therapy. Okay, I'd like to begin by mentioning that breast cancer is already the most diagnosed cancer in the world. It represents a serious threat to women's health because according to the statistics, it's estimated that by 2040, the incidence will increase 41% and the mortality by 52%. On the other side, the treatments for this pathology presenting disadvantage with respect to the efficacy and selectivity, development chemotherapy resistance, and endometrial cancer, as well as the lack of FDA-approved drugs for triple-negative breast cancer. Consequently, it is necessary to design strategies to identify drugs that target a particular activity. Nowadays, the estrogen receptor beta has been of increasing interest as an important pharmacological target in cancer. In this regard, the beta selective agonist darapropanetral or DPN, it has been demonstrated to inhibit the proliferation, increase the apoptosis in breast cancer cells, and even increase the expression of beta in cells with a down regulation, compared to the alpha activation that has opposite effects. And highlighting increased levels of beta result in a good prognosis and survival of patients with triple-negative breast cancer. Okay, considering this receptor as a therapeutic strategy, the following questions arises. Are there FDA-approved drugs that meet the structural agonist characteristics for beta activation? For the previous questions, it was necessary to explore the beta and the alpha binding sites because only are different in the next amino acids. For example, the methionine in beta receptor is replaced by a leucine in alpha receptor. And this isoleucine in beta receptor is replaced for a methionine in alpha receptors. Because of this, it was carried out a development of multi-complex based pharmacophore modeling for from 19 ligand protein structures of estrogen receptor beta. And here, I showed you the pharmacophoric map. It was created with 4 hydrophobic features, with 3 hydrogen bond donors, with 3 hydrogen bond acceptors. And subsequently, a massive exploration of FDA and drug bank libraries is exploring carry out using beta map. Additional exploration or additional virtual screening to exclude any alpha agonist was carried out using alpha map. Then virtual molecular docking simulations were used for optimize these results, and after a potential beta agonists were selected, in this case, were selected levodopa and sobetirone for having the necessary characteristics that are required in the beta maps, high pharmacophore fits, and better the binding energies in estrogen receptor beta in comparison with the estrogen receptor alpha. Subsequently, antiproliferative was performed in breast cancer cell lines, MCF7 and MDA-AB231, with a positive expression for this receptor, estrogen receptor beta. And for in vitro evaluation, both drugs had antiproliferative effects. In the case of sabetirone, the IC50 in MCF7 was 82 micromolar, And in MDAMB 231, the IC50 was 101 micromolar. And for leberolol, the IC50 in this cell line was 100 to 80 micromolar, to 8 micromolar. And in these cell lines, MDMB 231, the IC50 was 146 micromolar. Okay, and finally, we have the next conclusions. The application of multi-complex-based pharmacophore modeling of estrogen receptor beta allows the identification of drugs with high affinity for the beta receptor and antiproliferative activity in breast cancer cell lines. This work contributes with a viable alternative for the possible repositioning of sobetirone and leberolol to be used in the therapy against laminal breast cancer and aggressive triple negative. Thank you for your attention.
Thank you. And we have several questions. The first question from Pavel Polishuk. How did you create a single pharmacophore model from multiple complexes?
Okay, they create a single pharmacophore model for multiple complex. I would like to explain that by this slide. For example, we have each crystal proteins of each complex ligand proteins. Then in the software ligand is called software. We can align all the protein complex and then throw the share sphere shares in between all the complex are aligned, and the shares spheres we can refine the model.
Thank you. And from Kunal Roy, what is pharmacophore fit? How to evaluate quantitatively a pharmacophore?
Okay, there for evaluate the quantitatively a pharmacophore is, for example, that one of these spheres correspond with a score. A pharmacophore, for example, this value is the range to the one top the 100, and some ligands, for example, have 50, have 70, have 80, and we considering the score of the pharmacophore fit high valued when the score is higher, is 70 score in fit pharmacophore.
Yes, the question from Sonobeni, which software is used for pharmacophore modeling?
Okay, the software that we use for modeling pharmacophore, ligand is called software.
Yes, and from Vladimir Paroykov, are there known ERB agonists among the known drugs? If you validated the approach using these compounds, whether you validated the approach using these compounds?
Okay, yes, exists another beta agonist. In fact, we use this beta agonist to validate my pharmacophore model. For example, prinabetal, deripropernotriol, and other beta agonists reported in the clinic. And amongst that, these known drugs, we can validate my pharmacophore model. And another question, another situation important is that we using alpha agonists reported in the literature to validate the pharmacophore model.
Yes, and two questions from Dmitry Osolotkin. The first one, is IC50 on the level of 150 micromole relevant for real clinical use and further translation of these studies to clinics?
Okay, yes, the IC50 is relevant for real clinical use because, for example, another drug that is actually is today in repurposing is propranolol. For example, the IC50 reported for this drug is around 1000 in the range 200 micromolars in many cell lines. And the drugs that we consider for repurposing, for example, levodopa and sorbitol, is in this interval around between 200 and 100 micromolar.
Okay. Yes, and one more question. Did you measure receptor binding? What if the cell effect is realized through another mechanism?
Okay, the measure receptor binding, we not measure the binding, but another mechanism related is, for example, in the pathologies, for example, sorbitol is a neuroprotective and anti-sclerotic and anti-epidemic, and the studies is related in the receptor with the estrogen receptor beta and system nervous.
Okay, so thank you very much.